with aging, what happens is your ability to remove or repair the damaged mitochondria declines. And we see that quite evidently. We see it with things that improve this process of removal of damaged mitochondria, which is called mitophagy. They all decrease as you age. So it's not a surprise that you're not able to repair those as we age. So then what happens is you have all of these damaged mitochondria sticking around, hanging out in the cell and damaging the cell pretty majorly. So I think the flip side of mitochondria provide energy for the cell is not that you don't get enough energy. That's only one component. It's that you get a lot of damage that's actually caused by these damaged mitochondria. And that ends up being a bigger issue.
Hello, everybody. My name is Jose Navarro Betancourt, and I am the Scientific Director of quadraScope Ventures. We invest in early-stage healthcare companies that enable disease treatment and prevention through biological age reversal. And today, I will have a conversation with Spring Behrouz, the co-founder and CEO of one of her portfolio companies, Vincere Therapeutics. Spring, thank you very much for taking the time to speak with me.
Always a pleasure, Jose. Thank you for having me.
So let's dive in. What is Vincere's mission?
Our mission is actually quite broad. Our mission is to improve life, but then you sort of think about what is the best way to do that. And one of the biggest pain points, I think, in life for everybody is age-related decline, and particularly neurodegeneration, which is, I think, one of everybody's biggest fears. So that's the mission, is to improve life by combating the age-related decline that we see, particularly in neurodegenerative diseases.
Let's dive into the specific compound you're developing, and I specifically want to look at mitochondrial biology. How did you begin working in mitochondrial biology and in the whole mitophagy inducers? We will explain this later on.
Yeah, it's actually interesting. I've come back to mitochondria multiple times within my career. Even back when I was a PhD student, mitochondria sort of came up as an important hub, which is maybe not surprising. But for Vincere's purposes, we actually started trying to figure out what causes Parkinson's disease. So we looked at all of the genetic and other human data. We really wanted to start with human data, which should always be the case, but isn't. And we said, okay, what's the convergence? What are the things that all of the forms of Parkinson's disease, which is where we started, have in common? And that led us very, very strongly to mitochondria. So that was kind of the key thing that got us there. And then once we started working on mitochondria, that opened up a whole field. Mitochondria are one of the major hallmarks of aging. So then you have all of these diseases that have aging as the biggest risk factor, including Parkinson's disease, but also Alzheimer's, cancer, like so many cardiovascular disease, kidney disease, all of that now opened up.
What happens to mitochondria as we age? Why do we say that mitochondrial dysfunction is a hallmark of aging?
So, I think just in general, when I try to think about aging, I think what happens is that your repair machinery starts to not be as efficient, right? One thing that we have that machines don't have, for example, is the ability to repair ourselves when things go wrong. And that ability declines as we age. So we are less and less able, and you can see that very obviously, but like if you hurt yourself, it takes more time to heal, but it's also true at a cellular level. So when I think people think of mitochondria as, luckily, now everybody knows mitochondria are the powerhouses of the cell. They provide your cells with energy. But what most people don't know is that they're very dynamic. They're like tiny little bacteria. They move around your cells. They can, they can fission off of each other. They can fuse with each other. They get transported to the places they need to provide energy for, and they get damaged just like anything else in the cell. And with aging, what happens is your ability to remove or repair the damaged mitochondria declines. And we see that quite evidently. We see it with things that improve this process of removal of damaged mitochondria, which is called mitophagy. They all decrease as you age. So it's not a surprise that you're not able to repair those as we age. So then what happens is you have all of these damaged mitochondria sticking around, hanging out in the cell and damaging the cell pretty majorly. So I think the flip side of mitochondria provide energy for the cell is not that you don't get enough energy. That's only one component. It's that you get a lot of damage that's actually caused by these damaged mitochondria. And that ends up being a bigger issue for the cell when you have a lot of damaged mitochondria just spewing oxidative materials. And even if they get really damaged, they can burst. And mitochondria, just like bacteria, they have the circular DNA can get recognized by your immune system. Then your immune system is attacking itself. So it can cause a lot of different things. And that's all due to this. Well, it's not all due to this aging, but aging is a really big part of causing this.
People sometimes forget that one of the functions of mitochondria is they play a big role in regulating apoptosis or programmed cell death, right? So the mitochondria are a direct link between cellular dysfunction or mitochondrial dysfunction and then the induction of cell death. Let's talk about the key innovation behind Vincere's drugs. You mentioned two keywords, they are small molecules, and you also mentioned that they have to cross into the brain because you're targeting neurodegeneration or diseases of neurodegeneration. So what is this key innovation?
And we've been working on optimizing these molecules. So you really ultimately just need one molecule to go into the human, right? But in the process of trying to get that, getting a molecule that gets into the brain in high enough concentration to have the effect that you're looking for, that's a big challenge. I think that's one that most people don't realize. And we used to have these clinical trials that would show something, test the hypothesis of like, hey, this thing for Alzheimer's disease or for Parkinson's disease. And then at the end of the day, it was like, well, maybe it didn't get into the brain in high enough levels. So now I think everybody's, much more aware. And now we're trying to really design drugs that get into the brain, that actually do the thing that we want, in this case, enhance mitophagy in the brain. So that was part of what we did was, we use these really cool transgenic mice that have fluorescent mitochondria that change colors when they are being broken down. They go from green to red. And this is, piggybacking on a lot of innovation that's been done in the academic space. So one was developed by our collaborators, Ian Ganley at University of Dundee. One was developed by NIH, and Nuo Sun, who's now at Ohio State. And all of these are really allowing us to actually be able to see that we're getting into the brain before we go and test it in humans. And it's taken, 1,000 molecules for us to be able to optimize a molecule that has all of the right properties, that's safe, that gets into the brain, that doesn't get broken down in the system too quickly, and all of that. Yeah.
And I understand that there was a drug discovery engine that also led you to identify a particular target inside mitochondria. Can we please talk a little bit about that? Can you walk us through the target identification process?
Yeah, we were really kind of ahead of the curve on this because we used computational approaches to come up with the target, what target is the best one. So we used actually a variety of tools. I think people try to throw AI at everything, but AI may not, different types of AI may be good for different types of problems. So just kind of not treating everything as a nail. Sometimes you need a screwdriver, sometimes you need a hammer. So what we did was we used a suite of tools, and one of them was a physics-based simulation where we basically created a virtual three-dimensional cell, and you put in, based on human data, so you have human omics data, you can populate it with proteins and molecules and everything that's inside a normal cell, and you can do that for a healthy person or somebody with Parkinson's, somebody with Alzheimer's, and then you can see how the cells behave. And you can go in there and you can make manipulations very easily. In a human, of course, it takes many, many years to do that. In the simulation, you can do that very quickly. So that's where we came up with the target. We were able to alter USP30, which is our target, which is on the mitochondria, and see that we could reverse the abnormalities that we see in a Parkinson's cell and bring that back to a normal, get it to look like a normal cell. So that was really kind of a very key piece that gave us the confidence to go into this early. We were pretty early on it. And now, 5 years into developing these molecules, now we see a lot of other companies and, larger pharma companies being interested in this target. So the computational really gave us the conviction to go in early. And then we also used a lot of kind of analytics to look at the convergence. So we know, Parkinson's disease is nice in that there are a dozen or so genes that cause the disease. So we said, okay, what do these have in common? And we used a computational approach to basically hone in on what is the hub that is in common. And that's how we came up with mitochondria in the first place and mitophagy in the first place, which we then took into the simulation. Yeah, so quite a bit. I will say, though, there are some portions, and they're getting better now. But for example, like our medicinal chemistry team has done a wonderful job doing optimization of the small molecules without the help of a lot of computational approach on that front. So sometimes the human intelligence is still trumping the artificial intelligence.
So to summarize, you began using human data because humans first, and then you applied these computational tools to model these interactions between the small molecules and the mitochondria, and to see what would happen if you alter certain processes in the cell. And then this led you to look at USP30. You're optimizing this molecule. You chose small molecules because these molecules are, most drugs are small molecules. These are drugs that you can take by mouth, and we know how to, as humans, we know how to optimize them and tweak them and make them cross into the brain and all other sorts of things. And now, can you maybe tell us a little bit more about all the milestones you have achieved in preclinical disease models, like in mouse models of disease, basically?
Yeah. So what you just mentioned is, I think, a really key differentiation for us. We wanted to start with human, because the mouse models of Parkinson's disease or Alzheimer's disease, really any neurodegenerative disease, have not been predictive of what you would expect to see in humans. So a lot of things work in the mouse models, and then you go and test in humans, and there's no effect or very little effect in humans. And that's why we wanted to start with human data and really, looking at human as much as possible. Genetics, I think, that's one that has advanced very much and is kind of leading the field. But also then there's a ton of omics data that's now available, lipidomics, transcriptomics, proteomics. So lots and lots of data from human patients that have Parkinson's disease. In animal models, our focus has primarily been just to show safety, and also that the molecules get into the brain and they can enhance mitophagy in the brain. So those are kind of the key things that we focused on. And of course, the target has been shown to be protective in animal models of Parkinson's disease as well. So for example, in fruit flies that have genetic mutations that cause Parkinson's disease in humans, you see if you take out USP30, they're protected against neurodegeneration, and they're protected against motor deficits that come along with that as well. And then there was a recent Nature paper from a group at Harvard that showed the same thing in mouse models. So if you put in a protein, an aggregated protein, into the brain, you kind of get a Parkinsonian phenotype. And if you take out USP30, you're able to protect against that neurodegeneration quite a bit. It's quite a dramatic effect. So we definitely do see effects in animal models of the disease as well. But I think our unique approach is that we start with human data, and then, ultimately we want to test it in humans, but then along the way, you also test it in animal models. I hope that at some point we can just take out the animal models altogether, because frankly, it's not the disease, right? We're inducing the disease.
Perhaps with organoids, or perhaps with organs on a chip. Do you see that as a tool that eventually replaces animal models?
So I think there's still a little ways to go, particularly with organoids. One challenge with organoids, I guess it's the same challenge you have with people, which is, mice, for example, they're bred together to create kind of a homogeneous group that all behave the same. Humans are all very different from each other, right? And we saw this during COVID, like how different the responses were from human to the same virus. Some people died, some people hardly got affected. And it's the same, I think you have that challenge with organoids, because if you take it from one donor versus another donor, so there's a lot of variability. And there's the only way to overcome that is to have a large enough number. So I think that for things like organoids or organs on a chip, you just have to have, the cost has to be driven down enough that you can do a large number of biological samples from different people so that you can actually see the impact of your drug. But yeah, I hope this is where things go. And, some of those are pretty cool. There's like brain organoids are pretty neat. And some of them are, they're kind of scary, interesting.
Yeah, that's a whole different topic for another time.
We could discuss that for an hour.
Yeah, we could. So let's talk more about Parkinson's disease. I know Vincere recently received a $500,000 grant from the Michael J. Fox Foundation for Parkinson's research. Congratulations, by the way.
Thank you.
Why is mitophagy a more validated therapeutic target for Parkinson's compared to other neurodegenerative diseases?
Yeah, and I should give a shout out to the Michael J. Fox Foundation they've been actually supporters of us from the very beginning. They supported the computational approach when it was quite risky. As I said, this was, we started doing the computational work in 2014 when it wasn't really, as advanced as it is now. And they've continued to support us throughout this process, and hopefully will support us through into the clinic as well. Sorry, I lost your question.
No,
in my shout out.
Why is mitophagy a more validated target in Parkinson's compared to other diseases of neurodegeneration?
Yeah, so we know human genetics is really how we know the connection between mitophagy and Parkinson's disease. So if you have mutations in genes like PINK1 or Parkin, which are direct initiators of the mitophagy process, you get early onset forms of Parkinson's disease with high penetration, which means that the majority of people will get it. It's not just a risk factor that increases your risk for the disease. So we know that that there's a very direct link there. And then if you look at patients that have non-genetic forms of Parkinson's disease, which is the majority of patients, they all have deficits in mitophagy. So it seems pretty clear that in both genetic and non-genetic forms of Parkinson's disease, it has an effect. Now, that's not to say that it's not important for other neurodegenerative diseases. It's just that the human genetics makes it really clear case for Parkinson's disease. And I should mention, actually, there was one case of a duplication of USP30, a copy number variant. So you ended up with more USP30 alongside another gene. And that person ended up getting an early onset, very aggressive form of dementia, Alzheimer's. So I think there's definitely a link also to Alzheimer's disease. We see mitochondria being really important in Alzheimer's disease. There's nice evidence, and more of that is coming out. Some of it hasn't been published yet, so hopefully we'll see that being published. That mitochondrial dysfunction is actually a very early effect in Alzheimer's disease as well, before you end up seeing major degeneration. So I think that the most direct link is to Parkinson's disease because of that human genetics, but there's definitely a link to Alzheimer's disease as well.
And it all comes back to human data, right? To using human data to really decide where your research is going.
Yes, absolutely.
And let's talk a little bit about the downsides of this approach. So you're inhibiting this protein, this target USP30. What is the normal function of this protein? Why do we need it? Why is it there in the first place?
Yeah, that's an excellent question, and one that there's not a whole lot of answer to yet. I think we'll find out more. Generally, things that are not useful get sorted out through evolution. You don't need it. So I think that it does have a function. One of the functions that we know it has is it basically, the whole process of taking a mitochondria and breaking it down is very energetically consuming for the cell. So it's an expensive process for the cell, right? It's a lot of work. So what USP30 does is kind of shifts the threshold of, well, this isn't quite damaged enough to cause any damage to the cell. So let's just keep it around for a bit. It's still producing energy. It's still, providing its function. It's not so bad that it needs to be broken down. So it kind of errs on the side of, it's okay. We don't need to break it down, right? Because the cell has to make a binary decision. Do we take this mitochondria and break it down or not? There's no intermediate. So USB30 sort of says, let's not break it down. And so if you take out USB30, it then goes on the other side and says, okay, this one's damaged, let's break it down. Let's not risk it being, affecting the cell in any way. So I think there's that, shifting, and that may save your cells some energy and, a lot of expense that could be going towards other processes in the cell. But if you knock out or knock down USB30, we don't see any negative effect. So animals that have USB30 knocked out seem just fine. So it is an interesting question, but we haven't seen anything it does that would be essential, I guess, for the cells.
So I guess USB30 might have some redundant functions with other USB family proteins. And if you completely inhibit it, you're okay, basically.
Maybe some. It's interesting because it's the only USP that is only the ubiquitinating enzyme that is on the mitochondrial membrane. So it's the only one that's on the outer membrane. Now, there are some other USPs that are in the cytosol that can kind of complement as well, but not as well as USB30 does, because USB30 is always right there physically. So maybe you're right. Maybe some of the ones that are in the cytosol can kind of fill in for this one if it's not there. But we definitely see an effect when it's not there on mitophagy, right? It definitely shifts that threshold, but it needs the signal still. So this may be a reason why it's safe to take it out, is because if you take it out, it's kind of like taking your foot off the brakes. You still need the gas to go. So you still need the signal of mitophagy from usually pink one and Parkin. So it tends to be okay to take out USB30, but it definitely does shift that threshold of mitophagy. We see that. And we see it, primarily only when the mitochondria are damaged. We don't see it when the mitochondria are healthy. And I think that's why it's safer, is because we're only seeing the effect on damaged mitochondria. We don't really see any effect on healthy mitochondria.
Yeah, that's really interesting. This USB30 seems to be specific for damaged mitochondria then.
Yeah, exactly.
Very cool. So let's talk about the clinic. What key challenges have hindered the clinical development of mitochondria targeted therapies to date?
Yeah, I think there are a lot of different challenges for mitochondria specifically, but I think a general challenge that applies to all drug discovery is the time. And everybody always talks about the time and cost of drug discovery, but really it's the time. And it makes it really difficult to kind of do as a business model when it takes 20 years to test a hypothesis in human, right? So from the preclinical all the way to approval. So you have preclinical, phase 1, phase 2, phase 3 human clinical trials, and then approval by the FDA. The whole process is a 15 to 20 year time. And, you really, it's an entire person's career to do that. And then it might not work. So I think that is absolutely the biggest challenge. If we can compress the time of drug discovery, the cost will work itself out, because diseases like Alzheimer's or Parkinson's are such big challenges, and aging is such a big challenge, that if we could successfully answer the question in a year or two, instead of 15 years or 20 years, then we would be able to try many, many more things, even if it costs a lot of money. There's a lot of money in the world. I think time is a much, much bigger restriction. And, we're trying to actively think about how we can overcome that at every step of the process. And drug discovery is very inefficient. Every step of the process is very inefficient. So I think there's a lot of room for improvement. I hope a lot of people come into the space from the outside who have perspectives on how to get things to market faster. For example, in the tech industry, that happens very, very routinely.
When do you expect to begin clinical trials? What are the next milestones in this drug development process?
Yeah, we've just started doing some safety preclinical, pre-IND safety studies that are required by the FDA. So after that, we hope to start doing clinical trials. And our goal is to get this to patients as quickly as possible. I think the time we're very conscious of. So we want to definitely get this into human as quickly as we can. Started with human data. We want to get to human testing as quickly as we can. Of course, the main thing is just to make sure it's safe before we put it into human. That's what we're doing now.
These safety studies, you mean animals, right? Healthy animals.
Yep. So generally, the FDA requires, doing some animal testing before it can go into human, and that's what we're doing.
Would you consider conducting clinical trials in like alternative jurisdictions, say like Australia, Europe, the Middle East, the Caribbean?
Yeah, absolutely. I think the US FDA is one of the most stringent on as far as how much you have to show before you can go into humans. So Australia is actually one that is quite popular. Australia, Canada, even Europe sometimes. So we're considering that for sure, particularly for Australia. There's also a tax credit that's given to companies that do their phase 1 clinical trial, their first human clinical trial. So we're definitely thinking about that, and that could save us, 6 months to a year before we get to actually test it in humans. So it's a meaningful amount of time, I think. So yeah, we're definitely considering that.
Going back to the patients, what is the ideal patient population for this kind of intervention, for these mitophagy inducers? In which stage do you expect to make the most impact?
Always, I think the earlier you can go, the better. So we would be looking at early, very, very early stage, newly diagnosed Parkinson's patients. I think that would be our first set. We do think there is potential benefit for aging in general, but people age so differently that if your patient population or your test population is very similar to one another, it's best. So I think early onset Parkinson's disease, there actually are already efforts to try to diagnose people earlier before the onset of symptoms, right? And I think that would be even better. If you know somebody's going to get Parkinson's disease, but they haven't manifested with it yet, and you can intervene at that point, that would be better. However, I think those tools are not in place just yet. So likely for our trials, we will go after people who are newly diagnosed. And then hopefully, if we see really nice results, we can go into even people who haven't manifested with the disease yet, but are likely, very, very likely to get it.
So this second population where you would do more of a preventive or preventative approach, these you would really need very good biomarkers, obviously, correct?
Yes, absolutely. I'm glad you brought that up because biomarkers, I think, are a huge need. There's one that was recently, and this was a big effort by the Michael J. Fox Foundation and several different companies to come up with a biomarker for alpha-synuclein, which is a protein that aggregates in Parkinson's disease. So we at least have that now, and that tends to correlate pretty well with whether somebody will get Parkinson's disease or not. However, I think it's only one biomarker, and really what we need is a panel. So similar to when you go to the doctor and you do a lipid panel and they say, your HDL and your LDL and, your triglycerides are this, that's kind of what we need for mitochondria as well. So ideally, we don't even say, oh, you have Parkinson's disease, let me give you this mitophagy enhancing drug. We say, oh, you have mitophagy problems, let me give you this mitophagy enhancing drug. So that's the ideal case scenario with biomarkers that are actually very specific to the biological problem that we're addressing. And we're working on that too.
Let's dive a little bit into that. Like, are you actually trying to discover new biomarkers or trying to make a predictive model of how these biomarkers are related to the symptoms? What's your work on biomarkers?
Yeah, so the primary thing we're trying to do is just to show that our molecules are able to improve mitochondrial function or enhance mitophagy. And for brain, it's extra difficult because you don't have access to the brain. So you have to either do imaging or collect cerebrospinal fluid. But there's actually quite a bit of correlation between what you see in periphery, like blood, and what you see in the brain. So that's currently what we're looking is, taking basically a blood sample and being able to show that our small molecule medication can improve mitochondrial function. And then, of course, you can use the same thing to say, oh, this person is, deficient in mitochondrial function. And again, the way that we're doing that is through we try to do a kind of rational approach and say these are the mitochondrial things that we should be looking at, respiration or some other mitochondrial markers. But what we've seen so far, and this is still in development, so you're getting the the latest and greatest here from us, is just an unbiased approach. So what do we see change after we enhance mitophagy? And then we can sort of work our way backwards of how it's connected to our target. And that, I think that's been a bit more fruitful, because particularly for people who have good mitochondrial function, like a healthy individual, which is what you run your phase 1 clinical trial for, very little changes, right? Because we don't affect healthy mitochondria. So biomarker discovery is actually a whole challenge in itself, but I think it's so valuable. We talked about other mitochondrial approaches before, and I think we've talked about this before. I took NMN or NR supplements, and I had no idea, are they doing something? Are they not doing something? Who knows, right? And you're so biased. There's definitely a placebo effect in place where you think, oh, something good must be happening, but there's nothing definitive to show that. So that's what we're trying to overcome, is basically being able to definitively show that we're improving mitochondrial function with these biomarkers.
That sounds great. And obviously, as you said, you want to look at minimal invasive approaches. You want to look at the blood, and you also mentioned imaging. That would be even really cool. Obviously, with a mouse or with an animal, you can look at fluorescent mitochondria, but in humans, we don't have fluorescent mitochondria. So what would be like a good imaging approach for mitochondria?
Yeah, it's definitely a bigger challenge, and there are people who are trying to measure like oxygen consumption and things like that. I think that the biofluids, so like measuring things in the blood or even saliva, urine, other biofluids, is far more advanced than imaging efforts. And I think just because imaging is much more challenging, as you said, there are PET tracers, for example, that are being developed. Again, there's a PET tracer being developed by Merck for alpha-synuclein for Parkinson's disease, but I don't know of any that are in development for mitochondria specifically that really seem promising to me. I'm sure there are things in development. I hope maybe people will point them out after seeing this. But I think imaging is an extra challenge. But the nice thing about imaging is that you can actually, see it in the brain, see it in the part of the body that you're interested in. Yeah, but we're probably going the biofluids route ourselves.
Yeah, biofluids are also easily accessible.
Exactly. You don't need a device to measure it. It's much cheaper. It can be much cheaper, and you can measure it over time. So ideally, I think we tend to measure biomarkers very infrequently, even when we do, except for maybe glucose monitors where people are seeing that in real time. Now imagine if you can measure your mitochondrial function in real time. That would be pretty cool.
I understand that in these biofluids, most of the time, what you're actually measuring is the metabolites of mitochondrial function. You're not measuring the mitochondria themselves or how they work themselves. Is this the case? Is there like a trend towards looking at mitochondria directly, or are the metabolites enough?
I think so. Yes, both of those. As you said, most of the efforts are looking at mitochondrial metabolites. It would be cool. You certainly can look at mitochondrial respiration, but you have to look at that fresh. So you can't freeze the blood, for example, which means that if you go to, let's say, a Quest or hospital, they would have to have the assay right there. They would have to be able to do it right away. And it's not cheap. So those, for example, the seahorse assays that measure mitochondrial function are not cheap. So you really can't put one in every household for people to be able to measure in real time. And you could do it, and probably should. We should probably have assays where when you go to the doctor for your annual checkup, they measure your mitochondrial health as well. So I think that's certainly something that can be done. It's technically challenging to look at functional mitochondria because they're basically alive, right? So you can't freeze them. You can't do a lot with them. So this is why people look at the metabolites, because those you can freeze. You can even look at dried blood spots or things like that to measure some of the things. So it's just much easier. And some of them tend to correlate well, but you definitely miss a lot. You definitely miss a lot. Might be okay though. Similarly with cholesterol, you're going to miss a lot that's happening particularly if you're looking at cardiovascular health, but it still tends to correlate pretty well. So I think we take what we can at this point, and then we can continue to improve on it.
In the end, as you said, you're eventually going to use multiple biomarkers. It will never come down to a single thing. And so the more biomarkers you have a profile, and then you can make decisions based on that profile.
Exactly. And talking about the different profiles, earlier you asked about Parkinson's disease versus other neurodegeneration, for example. And I think it's likely that you have mitochondrial dysfunction in both, but maybe in slightly different ways, right? And so if you have a mitochondrial panel, it may actually be able to point you to which direction the abnormalities are, rather than just saying, oh, you have mitochondrial dysfunction, because mitochondria are really complicated organelles. So people just usually say, oh, yeah, you're going to improve mitochondrial health, but like, how are you going to improve it? And I think if you have a panel that can tell you exactly where the problem is, you can rationally go and fix that problem.
Yeah, mitochondrial panel. And what about the mitochondrial panel for other types of diseases beyond neurodegeneration? What other diseases are associated to mitochondrial dysfunction in addition to neurodegeneration related diseases?
Yeah, excellent question. And I'm a neuroscientist, so I tend to always talk about the brain, but it's really interesting so if you think about mitochondria being the energy producers, you can guess what are the other tissues, because the tissues that require a lot of energy besides your brain, your heart beating all the time, right? So the heart's one of them. Your kidneys that are filtering through all the fluid in your body. So that's definitely a big one. And we see with our molecules, very robust protection against kidney injury. And we see, for example, in heart as well, we see a reduction in cellular senescence when we induce accelerated aging, for example. So we definitely see improvement in heart and kidney function as well. I think the other one is skeletal muscle. So skeletal muscle, again, you need to lift things. So it's very energetically demanding tissue. We haven't looked much at that, but I would anticipate that would be another area that would be very heavily affected. And if you think about, what are the things that decline as you get older, all of the tissues that I just mentioned, right? Cardiovascular is a major cause of death. Kidney disease is a major cause of death. And you see people becoming frail and, old people not having really muscle mass. And then, of course, neurodegeneration. So this is why we say hallmark of aging, right? Because I think with just turning this one switch of improving mitochondrial health by removing the damaged ones, you can really affect multiple different tissues at the same time.
Very cool. So brain, heart, kidneys, and skeletal muscle.
Yep, those are the main ones. And then of course you remember that all of your cells have mitochondria. So, these are the main ones that we tend to think about, but I get actually requests of people wanting to put this on their skin because we know mitochondrial health is important for skin health as well. Of course, that's not where we're focused at the moment, but you can kind of see where people's interests are as well.
So we talked about mitochondrial function, but there's another dimension to this treatment, the autophagy parts. Dysfunction of autophagy is also considered a hallmark of aging. Obviously, stimulating autophagy is another therapeutic approach for these kind of age-related diseases. What is the rationale for specifically stimulating mitophagy versus general autophagy?
Well, I mean, again, I tend to be an optimist about approaches, and I think we should try many different approaches, particularly when it comes to aging, because it's something that affects everybody. So we should try as many different things as we can. So I will say I'm a fan of trying autophagy enhancement as well. But there are definitely differences. And one of the reasons that we chose mitophagy is because it is a very, very specialized form of autophagy. And you're not just removing, when you increase macroautophagy, you sort of just remove all the junk, I guess, but some not junk also at the same time. Whereas mitophagy, the way that we're enhancing it is we're only removing the damaged mitochondria. So it's a bit more rational, I guess I would say, in that you're only removing the pieces that are damaged. But I think that enhancing autophagy itself can have nice effects. There are things to worry about with enhancing autophagy too much, right? You can overactivate. Whereas at least with USP30, we don't seem to overactivate mitophagy. There are other ways you can overactivate mitophagy that could also be bad. You don't want to clear all of your healthy mitochondria, right? That could be really bad. So I would say that's one major advantage of going after mitophagy, is that you're only removing the damaged components and leaving the healthy ones alone. The other thing, which is super interesting and very recent advancement, is we seem to also remove like protein aggregates that tend to clump up around the mitochondria. So, these protein aggregates are very prominent in neurodegenerative diseases. And at least in the case of Parkinson's disease, where you have these abnormal phosphorylated forms of alpha-synuclein, which is a protein that aggregates, they tend to clump around mitochondria. And if you can remove the damaged mitochondria, you also remove these damaged proteins along with it. So it can be kind of a plus in two different ways, where you're very selectively removing only the parts that you want to remove, as opposed to just like, let's clean everything up. So I guess in that way, it's a very different approach.
So Vincere is developing mitophagy inducers. Mitophagy is now a validated drug discovery target. How does Vincere's approach stand out from the competition?
I think that the key thing is we've done rational drug design from human data. So starting with the human genetics and human omics data from patients, and then taking that and developing a drug that will ultimately go back into humans. Whereas I think a lot of other groups have, even if they have platforms, on that use some kind of assay that's been developed in the lab or animal model, or like developing for a whole set of targets that are similar, for example. And ours has been really very human-centric from the beginning, and then going back to human at the end.
So humans first. That's
humans first. Absolutely.
Mitophagy, as you know, has been gaining significant attention from big pharma, and there are a couple of interesting deals. The acquisition of Mitobridge by Astellas for $165 million in 2017. They were developing USP30 inhibitors. Then the acquisition of Mitokinin by AbbVie in a deal worth over $600 million in 2023. The Mitokinin was developing PINK1 targeted therapies. And then Vandria. Vandria is an emerging player in the space. Evolution invested in them. And actually, earlier this month, in December 2024, they began a phase 1 clinical trial on their lead asset. According to Vandria's publicly available IP, they are using urolithin A derivatives. So how does Vincere compare to these three companies?
In Mitobridge, they were kind of doing mitochondria before the field really got started. So kudos to them. But I think maybe they were a bit too far ahead of the field. I think they have quite a few things in the pipeline. They tend to, following their acquisition, they focused more on the clinical pipeline. And so their mitophagy enhancers actually didn't make that because they were quite early stage at that time. As far as Mitokinin is concerned, they are actually, a company that's I would say somewhat similar to ours. So it was really nice to see them actually successfully partner that with AbbVie. And, we have good friends at AbbVie as well. They're also very focused on neurodegeneration. So it's really nice to see, as you say, the enthusiasm pick up for this target, because when we started, it was like, it was pretty immature still. So the space is definitely starting to mature, which is really nice to see. Mitochondria is focused on PINK1, so it's a different target, but it is also a rational target. It is exactly involved in the same process that we're talking about now. There may be potential for, complementary. We could combine the two even potentially. One thing is, if you have a patient that has a mutation, for example, in PINK1 or Parkin, probably our approach would be better than their approach, but it's directly genetically linked to Parkinson's disease. So again, I tend to be rather optimistic about it and rooting for that approach as well. Vandria, as you said, they're doing a urolithin A analog. And I'm Persian. We eat a lot of pomegranates, so I'm always I'm always partial to eating pomegranates, and it's got lots of good things for you. And that's a kind of big source of ellagic acid and urolithin A. But I think one challenge with urolithin A, but there are a couple of challenges with urolithin A. One is that we don't exactly understand the mechanism. So how is it enhancing mitophagy? It seems to maybe be enhancing mitophagy by depolarizing the mitochondria a little bit. So causing a little bit of damage that then the cell says, oh, we should get rid of this, right? This damaged enough. Whereas ours is just kind of artificially changing the threshold to say, remove the mitochondria that are kind of damaged. This one is maybe pushing them to be a little more damaged so they get cleared by the cell. It might be an effective strategy, but I do specifically wonder, I think it would be better if we can remove them without causing damage to the mitochondria. So we kind of still like our approach better than that, better than causing damage to remove them. The second thing is the dose. I'm just not sure. It seems like you really need a high dose of urolithin A. So I don't know how much more potent these analogs are. But that would be a big concern, is like, are you getting enough of it particularly in the brain for something like Alzheimer's, which is what they're going after. And one other thing, again, I tend to be on the optimistic side, but they're doing their phase 1 in males only. I'm not sure what the rationale is on that, particularly because Alzheimer's disease is more common in women than men. So I want to call them out on that one. But I think that's a very common and unfortunate thing that we tend to test only in one sex, and particularly in a disease that's more common in the other. I hope that they consider like adding, women to their phase 1 trial as well.
Yeah, that's a really interesting point, and I guess only time will tell.
Yes, yeah, but I hope that they work. Again, urolithin, I think, is there are supplements already that you can take because it's a natural substance, right? So you can take, and I should say, I took urolithin for some time, and again, hard to tell because we have no biomarkers. It's hard to tell in a healthy individual if it's doing anything or not. So again, and I don't think, as far as I know, I don't know if they're looking at any specific biomarkers, but that would be really, really cool if they can look at biomarkers and actually show that the compound is doing something useful.
Is this a supplement, the Mitopure? I think
yes.
That's
Yep.
urolithin.
And again, I guess I'm not advocating for anything. I usually tend to try them. I've tried NR, NMN, urolithin. And it's kind of hard to tell if anything is happening or not, just because we have nothing to measure it. But I like the idea.
So, Spring, how do you see these mitophagy inducers working together with other mitochondria-targeted therapies? For example, with mitochondrial transplantation, which has gained some traction recently.
Yeah, mitochondrial transplantation it's really cool, and it's one of those things that you just don't think is going to work, right? Basically, you're taking mitochondria and you're injecting it in. I think it would be the two could really work very much synergistically together, because if you're putting in a whole lot of new mitochondria, a lot of them are going to probably be damaged. As you're processing them, you're putting them in a tube, you're spinning them down, you're going to inject them back in. So I think having mitophagy, enhanced mitophagy as part of that will be essential to remove the damaged ones, because you don't want to have a whole lot of damaged mitochondria hanging around. So I think it would be a really cool complement actually to have more mitochondria, maybe through transplantation, and then remove the bad ones out of the system. One thing I wonder about is for brain, making sure you actually are getting the mitochondria into the brain. It's always a big challenge. With small molecules, you can make sure that they're very small and lipophilic. Mitochondria, of course, are big organelles. So can you get them in the brain? And it's really super interesting because your body, we don't think about mitochondria going in and out of cells, but they do. I mean, there's some evidence for mitochondria going in and out of cells. So that's really interesting to see. I would actually be very interested to see how it works for central nervous system, like the brain specifically. But again, I think you're going to need to, it would be good to pair it with something that removes the damaged mitochondria out of the system if you're going to be injecting a whole bunch of mitochondria into the system.
You mentioned that mitochondrial transplantation happens in healthy cells. Cells do that normally in the body. But a thing that I wonder, and that many people wonder, is, are these cells that are transferring mitochondria between each other using the mitochondria as an energy resource? Are they sharing energy, or are they using mitochondria as a communication device? Right? Is this only like a signaling phenomenon rather than like an energy transfer thing? I think that still remains to be seen, and I don't have a clear answer for that, and I don't know if there is.
Yeah, there isn't an answer yet for that. It's under investigation. It's definitely a really interesting area. I mean, I would be surprised if it's not somewhat for signaling purposes, because that's how cells communicate with each other all the time, and in more than just neurotransmitters and things that we're sort of used to. We're seeing all kinds of things go back and forth that were previously not thought to go back and forth, including mitochondria or like mitochondrial derived vesicles or components of. So I think there's definitely some signaling, whether it's sharing of energy or not. One thing that always was counterintuitive to me when I was studying neuroscience is you have these cells that have very long axons, and the lysosomes are usually in the cell body region. You get some in the axons and the axon terminal regions, but it's primarily, you learn lysosomes are in the cell body. Mitochondria are everywhere. Mm-hmm. So if you think about, well, if you want to break down this mitochondria, you have to ship it all the way back to the cell body, in some cases, quite a long ways. Or you could just send it to your neighboring microglia or whatever and have it be broken down. And now we're just talking completely theoretical here. None of this has been proven, but it sort of logically makes sense that you would maybe transfer that to a neighboring cell to be broken down rather than try to ship it all the way back to the cell body to be broken down, especially if those neighboring cells are cells that are have the machinery to do that easily. So I think some of it is, and to go back to your question, I don't know how much of it is actually energy transfer as opposed to signaling, or even for degradation purposes. Maybe you want to get rid of this, but you don't want to send it all the way back.
So it seems Parkinson's is specifically a good target to be treated by cell therapy because it's caused by the loss of a very particular type of brain cell, right? So how do you see these mitophagy inducers being used in combination with regenerative approaches?
Yeah, I think that they have, because it's such a complementary approach, I think there's really good synergy, as you said. For stem cells, I think enhancing mitophagy may actually allow the stem cells to survive more. So one of the big challenges with stem cell therapy is you lose so many of them as soon as you put them in, right? The great majority of the stem cells die. So combining with mitophagy enhancers may actually allow more of them to stay alive. And, you have to think about the fact that you're also putting those cells into a very hostile environment. There's a reason that the cells degenerate in the first place. So the environment is not a happy place where these poor stem cells are being dropped off. so to speak, and they have to grow, they have to grow axons and terminals and project them to the right place. It's sort of amazing that that whole field even works in any way. I'm continuously impressed. So I think definitely this can be synergistic with that to allow the mitochondria to be healthier, to allow the environment to be healthier where the stem cells are being implanted. And just in general, we know that these neurons degenerate, and mitochondrial function is a big component of that. So, keeping them from degenerating. There's also a lot of potential with other types of therapies. I think if you're talking about Parkinson's disease, for example, there are approaches that are going after protein aggregation, like alpha-synuclein. Now, if you combine this with mitochondrial function, also enhance mitochondrial function and remove the aggregates, you may have much better effect on neurodegeneration than if you're just doing one or the other alone. So there's tons of stuff that you can do with aging as well. Like what if you're enhancing DNA repair that probably it's going to have an impact on mitochondrial health anyway, because a lot of the mitochondrial genes are encoded in the DNA. But then if you improve mitochondrial health, that also improves DNA health, because mitochondria are providing a lot of the energy and also provide a lot of the things that damage the cells, the nucleus. So lots of interconnection between those. So I think mitochondrial therapies can be potentially complementary to a lot of the other approaches that are around. So let's get it to work first, and then we can do combination therapies with a bunch of different things.
That's right. Let's get it to work first. And as you said, everything in the cell is all connected.
But I think what's exciting is that the possibilities are sort of endless. We can think of many, many things to do once we have a pill that a person can take that actually improves mitochondrial function.
So my last question is, what is the best advice you have ever received?
I think the best advice that I've ever received was actually not specifically given to me. It was given to, I think Bill Gates said it to I forget which of the new CEOs coming in was Ballmer or Vijay, but he said that don't try to be somebody else. You have to try to do things as you. And I think as a startup founder, you're always looking to see what everybody else has done. And that sort of tends to limit you. We tend to think, oh, well, what is, what's AbbVie doing? What's Biogen doing? What's Merck doing? And I think that can be very limiting. I think you have to sit down and go, here's the problem. How would I best solve this problem? How would my team best solve this problem, without looking at what everybody else is doing first? So I think that's been probably the best piece of advice that I've been given. And then having conviction in that. We started with the computational work and everybody told us we were crazy. And here we are now. So it clearly worked out.
That's great. Thank you for sharing that advice, Spring. And thanks again
thank Bill Gates for that one.
and thanks again for taking the time to speak with me. It was really interesting. It was really fun.
Likewise. Thank you, Jose. Always a pleasure.
Thanks. Hello again. If you found this video interesting and would like more people to see this kind of content, consider clicking the like button and subscribing to our channel. Thank you for listening.